All right. One last time. What are we going to begin with?
Last lesson.
What? The first lesson, immunology. So inflammation and phagocytosis. What do we want to do with it? All right. Okay. Here's where we'll start. For those of you that want to start with this, let's say steps of inflammation. Okay, so we'll do 3 different problems to get us going. One will be steps of inflammation, and you must use the following words in your steps properly. Margination. I probably spelled that wrong. Diathesis. Hey, get using the words. Cytokines, chemokines. Neutrophil. What else can we include to this list? Write out the steps of inflammation or describe inflammation using this terminology. Oh, it is, but there's more. Let's throw macrophage in there. Okay, so that'll be our first list of terms. Oh, why? No, you know what? We're going to throw in one more. 2 more, 3 more. PRR, PAMP. Phagocytosis. Because this is all part of inflammation in some ways too. All right? So, your job to get us going, first 5-ish minutes or so. Steps of inflammation, what's happening during inflammation. Use these terms in your response. Okay? And then I'm going to write some more terms so that once you're done with this first part, you can start to do some more terminology bingo. Hey, Danica. Yes, I am. I just haven't gotten to it yet, but I will post that. I write it in my hand so I don't forget. Go for it.
And the Wi-Fi kept cutting out, so that's it was definitely a bad one to not do really good. Yeah, the basis of everything. Okay.
It's not on there. It's just
The macrophage is coming later. You do this for everything.
yeah.
So I just take notes. Oh, I need a book? Yeah.
No, but I.
For the second one, I'd like you to start creating a concept map. Oh, you don't have to. Nobody's forcing you to. I'm just saying I'd like you to. Yeah, well, I don't like other things either.
Okay, and you're like it's
Oh, and then something that we've talked about. Okay, let's go back to lac operon for a minute. I know, everybody's favorite. I know. When lactose binds to the lac operator, what happens to the to the inhibitor? What happens to the inhibitor? Yeah. What happens to the inhibitor itself? Abby?
It comes off and allows them to.
Changes shape, comes off, right? Think about another example. When dopamine, when dopamine binds to a neuron, what happens? I know some of you are covering brain stuff right now, I think. It stimulates the brain. How? Okay. It sends signals, but it does so by binding to a receptor on the cell surface. It binds to the dopamine receptor. Okay? So the question is, how do cytokines work? Cytokines are secreted molecules. They function by binding to the surface of any of the immune cells. And we're just describing this in general. They can bind to any immune cell. And by binding to that immune cell, it changes the immune cell's function, or it changes its activity. We've said a few times now that cytokines are required to activate B cells, or are required to activate a TC cell. And the way that happens is that once the ligand, in this case, the cytokine binds to the receptor on the cell surface, it's as if it switches, it turns on the cell. It takes it from like this resting dormant state to now an active state. So it's kind of like a light switch. That's what a cytokine is. Does that help, Emma?
Yeah.
Okay.
Turns on inflammation response.
It turns on inflammation response. Exactly. So cytokines have many roles. We have just basically described, like, here are general things cytokines do. They activate the immune cells. They turn on or activate inflammation or stimulate inflammation. There's a lot more, there are more caveats and depth than just that, but that's all I'm asking you guys to know. They're activators of the immune response. Okay? All right. So how do we use these terms? And let's just start anywhere, and then what we'll do is we'll build this up. So I'm going to start a new slide here, and we'll just kind of build this up from any place you guys want to begin with this. So someone give me a starting place. What?
Tissue damage.
Tissue damage. Wonderful. Yep. Okay, so injury. What happens next? Cytokines don't get released first. Something has to happen in order for cytokines to be released. Allison? Okay. PAMP binds PRR. Which of these is found on the pathogen? PAMP, which is found on the macrophage? The PRR. Which one is found on the B cell? None of them. All right. So PAMP binds PRR. What happens after this event? Phagocytosis, he says with a mouthful of chicken fingers. Phagocytosis. And let's just use this opportunity then to walk through our steps of phagocytosis. So step 1, PRR binds to PAMP, internalizes, forms this endocytic vesicle known as a phagosome. What's the next step? What happens to that phagosome? Gets more acidic. It matures is the other term we used. After that, Fuses with the lysosome to form the phagolysosome, and what happens in the phagolysosome? Pathogen is killed. Because what is being released in this phagolysosome? What was that, Erica? Reactive oxygen species, reactive nitrogen species, which will oxidize the organism, disrupt the macromolecular structures, including protein and DNA, lead to the death of the organism. At that point, what happens? If this is a macrophage, what happens after this process of phagocytosis? If it's in the phagolysosome, Derek, which of those two? No, that's fine. You don't need to apologize. We're just going through this right now. So MHC2, remember, is the one that is used, we think of, when there's a pathogen from the external environment being brought into the cell and then being presented. It's on MHC2. So one thing that can happen after phagocytosis is presentation. I ran out of room. What's the other thing that could happen? Or I should say, what also happens at the exact same time? What does this macrophage begin to secrete? Cytokines. What do this? Go ahead, Mark. Nice. Cytokine, I'll put this in parentheses here, and chemokines. And I put it in parentheses because chemokines are a specific type of cytokine. What is the function of a chemokine? It is attraction. It is a chemoattractant, right? So it's involved in chemotaxis and recruiting something to that inflammation site. Danica.
Will you repeat what presentation
What that actually is. Can we come back to it? We will come back to it. That's why I've drawn it over here so that we can draw this arrow that's going to actually lead to the next slide. We'll come back to it. All right. So cytokines, chemokines secreted. What now happens? What's that? Not just yet. A couple of things have to happen before margination can occur. Meg? So vasodilation, that's going to be a result of the cytokines. Increases blood flow. Derek?
The CD8 and the TCR on cytotoxic T cells detect the MHC2.
So we'll come back to that. For TC, it's actually MHC1 in those cases. So it's the T helper is MHC2. I'm just going to write a couple of these terms over here. T helper. MHC2. Okay, we'll come back to that. And what is edema? Swelling. And particularly, the swelling is caused by what? What's happening to lead to that swelling? Meg? Yeah, so the plasma from the blood vessels is now moving out of the vessels and into the tissue. So not only is there vasodilation occurring, but we start to get leaky blood vessels too. We start to get a little bit of separation from the endothelial cells that line that blood vessel, now allowing for this tissue to move into that site of where the wound has occurred. There is a benefit to this. This is not just a side effect. We never talked about what the benefit is. Okay, and we will not talk about it. It will be way too confusing. What else is happening? Who comes to the party? Who's the one that shows up to the party right on time? Neutrophil. And the one that stays too late. Neutrophil joins the party. How is he or she recruited? Chemokines. Recruit that neutrophil. So neutrophil recruited by chemokines. So the neutrophils are, in this process, just kind of, here's our blood vessel. The neutrophils are just kind of like bounding along, and then all of a sudden they sense chemokine and they change. Again, chemokines, cytokines, they activate responses. They activate the immune response. And this neutrophil goes from a bounder to all of a sudden it just kind of flattens out and grabs onto the cell wall. Or sorry, to the endothelial wall. Benefit of this, it prevents that neutrophil from just kind of like rolling past the site of inflammation. Because blood flow is in only one direction. It's not as if a cell can back up in the opposite direction of blood flow. So what the neutrophil does is it grabs onto the surface or to those endothelial cells. That stops its movement or slows it down so that now it can move more slowly, more carefully towards the site of where chemokine concentration is the highest. Once it reaches that site, oh, and what is this process called? This is our margination. And eventually, what happens? Goes into the tissue, process known as diapedesis. Go ahead, Mike.
So is the margination by the neutrophils or by the macrophages?
That's a great question. As we're talking about it right now, neutrophils. But one of the things that we need to remind ourselves of is inflammation goes beyond just these initial steps. It continues. When neutrophils begin to induce phagocytosis, this process actually occurs all over again, or increases or intensifies, where now instead of neutrophils being recruited, it's going to be macrophages. So the answer to your question, Meg, is it's both. The macrophages would go through the same exact process of margination and the other term, diapedesis, to be brought to the site of infection. You can actually age an infection by the types of white blood cells that are located there. If it's an early infection, neutrophils. If it's a later infection, macrophages. Erica. Yeah, it's a different set of chemokines. Again, we're using very general terms here, but yeah, it is chemokines that would be recruiting them. What terms have we not used? Oh, son of a monkey's uncle. The words in my head are a lot worse than that, though. Huh. Oh, okay. Let's see. What have we not? I think we've used all of those up there. Margination, diapedesis. What was that? Yeah, we did. PRR is on there. PAMP binds to PRR. What is a PAMP? What's the acronym stand for? Pathogen-associated molecular pattern. PRR, which I could never figure out for some reason. Pattern recognition receptor. Devin. So this Meg was asking, do macrophages when macrophages are recruited, are they undergoing margination diapedesis? And that what I'm trying to model here is the neutrophils, once they are recruited to that site where the injury has occurred, they will begin phagocytosis as well. As they are performing phagocytosis, they will secrete cytokines and chemokines that will begin to recruit macrophages to that site of infection. Yes, exactly. Macrophages are the late arrivers to the party.
Do they always have to show up?
Do macrophages always have to show up? Most of it?
I'm just curious.
I assume that macrophages do show up because neutrophils in the process of the phagocytosis would be secreting these chemokines and cytokines. Does that mean the macrophages are going to continue phagocytosis? Maybe that they show up and everything's done, and their job at that point is to just clean up debris, dead cells, etc. Yeah, it's a good question. Sydney.
Can you explain diabetes again?
Yeah. So the diapedesis here is once that, in this case, well, I'll just describe it for a neutrophil. Once that neutrophil marginates or grabs onto the wall, it's going to start moving towards higher and higher concentrations of chemokine. Where is that chemokine at its highest concentration? So let's say, I'll go to a next slide because I need more room to draw. So blood vessel, marginating neutrophil, wound, bacteria, macrophage. Where is the concentration of the chemokine highest? At the site of the tissue damage. And so what happens is at some point in the blood vessel, the concentration of chemokines no longer increases. The only way for the neutrophil to continue moving towards higher concentrations is to actually move out of the blood vessel and into the tissue. That's the diapedesis. So the diapedesis is really referring to this process in which that neutrophil will, this would be like the two ends of two endothelial cells. The neutrophil is actually squeezing its way in between those two cells to get into now the site where the infection is occurring. So the diapedesis is the movement of the neutrophils or macrophages from the blood vessels into the tissue. Good question. Inflammation would occur if I want to answer this in a couple of different ways. One is to say inflammation would occur if a macrophage recognizes a PAMP. That's going to begin this process of inflammation. There is also, though, let's say it's a sterile knife. There is going to be there will be times in which that causes tissue damage, but where maybe that doesn't necessarily immediately trigger an inflammation response. But if you think about an open wound, that's actually, one term we did not talk about is like first, second, and third line of defenses. Your skin constitutes what we call a first line of defense. If it is broken, then now you've got cells, blood vessels, etc., where you have created a portal of entry for bacteria, virus, etc., to attach to. It's probably at that point where you'd start to see the inflammation occur, once the macrophages come into contact with those. The additional damage, one of the other roles of macrophages is to help phagocytize dead necrotic cells as well. So those necrotizing cells would probably trigger macrophages to be recruited. I'm not sure so much about neutrophils. I don't know how the response would necessarily differ. That's a great question, though. Emma?
So phagocytosis is always part of inflammation.
Yes. Yeah, and phagocytosis really is kind of like the initiator in some ways because that initial binding of the PRR to the PAMP is what's going to trigger the secretion or production of cytokines and secretion of cytokines. Okay, other questions on the process of inflammation, and we've kind of covered phagocytosis in brief terms too. Kaylee? So the macrophage, there's 2 points at which we observe macrophages in the inflammation process. There are Stationary macrophages, ones that are already at the site of where the infection is occurring. So I guess I'll go back to my original picture. This would be an example of a stationary macrophage. So they're kind of like embedded in the tissue. And then there are others that after the neutrophils make their way to that site of infection and begin to secrete cytokines, then there are additional macrophages that are in the bloodstream. So these are ones that are mobile, and they would eventually be recruited to that site of infection as well. Chemokines secreted by the neutrophils. So in the same way that neutrophils get recruited by macrophages. How about these ones right? So the ones that are stationary are already there. You've already got them in your tissue. We have macrophages that lay just below the surface of the basal epithelium so that it can protect you against any cuts or wounds. We have ones in the lungs that are there to protect against any pathogens that make it through the ciliary escalator and make its way into the lungs as well. And anytime their activation signal is really the binding to the pathogen. That's their activation signal. Yeah. Devin and then Meg.
So there's neutrophils on the outside within the tissue, and there's neutrophils within the blood vessels.
There are macrophages in the tissue already there.
How long? It's like neutrophils the only ones that can secrete the cytokine.
Well, both can. Both can secrete cytokines, but and both do when they bind to a pathogen. The timing is different of when it happens, right? So the macrophage, there are stationary macrophages there first. They recruit neutrophils. The neutrophils, once they are recruited, will help to recruit more macrophages to that site of infection as well. Does that help, Devin?
Yeah.
Okay. Meg.
Phagocytosis, and you were saying that MHC2 is inside of the cell, and then like puts itself on the outside. What did you mean by that?
Let's
How
go
do
to
you know
that.
It does that, and MHC1 doesn't.
Okay. How do I know that
How does
MHC?
that happen? Or like, why does MHC2 do that and not MHC?
The simple answer is because I told you so. The long answer is a bit more complex than that and involves basically a discussion about where do these molecules localize once they are produced in the cell. So MHC2 oftentimes is being budded off from the endoplasmic reticulum actually, and it will fuse in these endocytic vesicles. They're going to fuse with phagolysosomes. In the case of what's that?
So it like takes a piece of the pathogen and then like puts that on the outside of it?
Yeah. So what happens is, and maybe that takes us to this conversation here on the interaction of these different molecules. But I can try to draw this out a little bit real quick. I'm going to go forward a couple of slides. So here's our macrophage. Okay. Let's assume that the macrophage has gone through the process of its PRR has bound to some PAMP on a pathogen, has internalized it, phagocytized it, lysed it, and now we've got these bits and pieces of pathogen. Okay. From the endoplasmic reticulum, we've got other vesicles carrying MHC2. And these will fuse together. So we'll get a fusion event between these two vesicles. Yep. This is presentation, so after phagocytosis. So fusion of those two vesicles. MHC2 at this point now has all of these different pieces of the pathogen or these antigens that could bind to it. And it's kind of random chance which one gets chosen, Meg.
Okay.
once that binding has occurred, this whole vesicle actually will move its way to the cell surface. And as it's moving to the surface, I'm going to extend this out a little bit. What we're going to find is not only does the vesicle here become part of the cell's membrane itself as this kind of expands, but we're going to release the additional antigen into the environment or into the tissue, and MHC will eventually become presented on the surface. Okay, so that's the MHC2 presentation. And again, the way to describe this is we've got an extracellular pathogen, one that's being brought from the outside of the cell, broken down once inside of the cell, and then being presented back on the outside surface. What cells can perform this particular type of presentation? Well, MHC2 is the molecule that's involved. And so the other way to ask this is, which cells are the only ones to express MHC2?
Macrophages, B cells.
Perfect, Zach. Macrophages and B cells. So B, macros. What's another term to define these two cells?
PACs.
Professional antigen presenting cells. Go ahead, Kaylin.
Doesn't MHC2 also need T helper cells to activate B cells?
It acted, yes, and so it's not that the T helper expresses MHC2. The T helper binds MHC2
To express the B.
so that the B cell can become activated. Yes. So we'll come back to that a little bit. Meg,
So what's the difference between this and then MHC1?
that's a great question. Should we segue into that? Everybody okay? Okay. So MHC1 is there we go. MHC1 is a tad different. MHC1 requires an intracellular pathogen, or one that is actually producing protein inside of the host cell. In other words, MHC1 is really good for what type of pathogen? What in particular do we know has to replicate inside of a host cell? Virus and? I'm looking at you two in particular because you did a presentation on it. Chlamydia, yes. All right. So, any other cell has MHC1. All right. So what we have to have is, in this case, I'll draw a happy virus.
All cells have MHC1, you said?
All cells have MHC1. All cells with a nucleus have MHC1. So here's a happy virus. It undergoes the process of replication, transcription, translation, begins to produce new pieces of virus. Yeah. And at all times inside our cells, there is, call it cannibalism if you want to. There's always this constant like flavoring of what's being produced. Okay? So what's going to happen is these proteins in here are going to be broken down by this garbage can looking structure known as the proteasome.
Is that something you should know?
Not really. No, I'm just giving context. Better to say proteasome than to say the garbage can inside of our cells, probably. So pieces of virus go in. They get broken down into even smaller pieces. These eventually, by a process that we will not cover because of the complexity, will bind to MHC1. And the MHC1 is actually found on the endoplasmic reticulum in this case. So eventually what we will find is endocytic vesicles with MHC1 containing bits of this broken down pathogen. And in the same way, this gets exocytosed to the surface of the cell, so that ultimately what we will, wrong color. Ultimately what we will get is a cell studded with MHC1. And each of these MHC1 might be presenting a different piece of the pathogen, or a piece of itself, its own cell. And any of those can then be bound by T cell receptors and TC cells. Yes, yes. So we'll come back to this and we'll concept map a little bit, or organize it. TC cells, well, yes, we haven't mentioned that, but you're absolutely right on K cells as well.
So the TC cells recognize them as a pathogen?
Yeah, so there are, again, this is not something that we're going to talk about because of how complex immunity is. There's a reason why there is a course that we have called immunology, and it's because you can take this and talk for at least a semester, if not a year, about the complexity of this. But the T cells have been trained to recognize or distinguish self from foreign. So if it recognizes self, it doesn't do anything to the cell. If it recognizes it as foreign, that activates it.
And it's foreign because it's that MHC1 with a part of the virus.
Yes. Yeah, it's actually that part of the virus which is considered to be foreign. Yeah.
How does the NK cells respond?
Should we go over how these two cells respond differently from each other? Compare. It's a great question, Sydney. Hint. Spend some time with it. All right.
Isn't there something about like the tail of something or toe of something?
Say that again?
There's something about like the tail of something that
Maybe.
like
Oh, toll-like receptor? That's a PRR. It's a specific type of PRR. You're welcome.
Yeah.
Yes.
So the MH1, like the receptors come out, but the cell's studded because of MH1.
So, the term that I used is studded, but basically you've got MHC1 presented on every single cell. And so it is basically a way, as this cell is constantly communicating with everything around it to say if it's normal or if there's something wrong
And
with
so
it.
then the TC cells and the T cells bind to it, and if they see self, then they just like.
Then they just they come, they attach, and then they move away.
But it doesn't say how to
Yeah. If they notice foreign, that becomes the next step here. Then they kill. All right. So let's say we have. I drew this wrong. Let me. Two SAD cells. One presents MHC1. Another one does not. Let's say a virus is inhibiting its ability to present it. It's supposed to be teeth. Okay. In the cell that is presenting MHC1, so the cell on the right here, what will bind to it? TC cell. T cytotoxic. So we'll draw this guy in red. Only if it's foreign. We'll assume that this is a foreign one. No, that's fair enough. Fair enough. And the TC cell has two receptors that we talked about.
Is that the CD8 and CD4?
CD8 is on the TC. So that's what we would call a co-receptor. So the CD8 is a co-receptor that binds MHC1. In other words, the T cell doesn't function unless this antigen or unless this part of the pathogen is actually in MHC1. Okay? And this is really important because what is the function of a TC cell? What does the TC cell do? What's that, Cindy?
Recognize if it's part of the cell or if it's foreign part.
Let's assume it's foreign. What happens then? Binds. There are specific things. That is exactly true. I am looking for two words to define what this thing secretes. Granzymes and perforin. What do those do? They kill it. They lyse that, or they don't lyse it. They just trigger this cell to die. Okay? So that TC cell, when CD8 binds to MHC1 and when the T cell receptor binds to the antigen, this here is that T cell receptor. What that is going to do is to force this T cell to now secrete from it those granzymes and perforin.
Hold on. So, what's the difference between TCR and CD8?
So the TCR is the T cell receptor. It recognizes the antigen. CD8 is the co-receptor. It recognizes MHC1. All right, so those two events have to happen at the exact same time in order for this T cell to secrete perforin and granzyme.
So this CD8 helps it bind the MHC1 to bind to with the T.
Yeah, and this is really important. And the reason why is because we're not showing this in the image, but the contact between these two cells, the TC cell, the cytotoxic T cell, and that infected cell, it's very close. In other words, the granzymes and the perforin are basically going to go from one cell to another over a very short period of space. And this should, I hope that this becomes apparent why this is important. What if a T cell is out in the middle of nowhere or out and surrounded by cells? It recognizes the antigen and then just starts shooting off perforin, granzymes. It kills other cells that it doesn't intend to. It's collateral damage. So that's why these two things have to happen at the same time. It ensures that you've got close contact and that the only cell it's going to kill is the infected cell. Kaylin.
Is that where immunosuppressant comes into play? It's like getting enough more cells in the periphery?
Yeah, so we have autoimmune diseases where those TC cells might be killing additional cells. Sometimes it's they don't recognize their self as being self. They recognize it as foreign. You get killing of other cells erroneously. So that's where you might put somebody on an immunosuppressant. Great case in point, transplantation. It's foreign. So you get TC cells and NK cells that might be active, not recognize that MHC1, and trigger death of that transplant.
So then you're being put on an immunosuppression, but then to go after the immunosuppression versus the organ?
So you put them on an immunosuppressant so that the TC cells are basically, how do I want to describe this?
So they go basically inactive, so they don't.
Yeah, that's the best way to describe it. Is they go into this more, or it takes a lot more to activate them, so that they aren't just willy-nilly killing off the transplant. Sydney.
So you said the CD8 and at the same time.
Simultaneously, exactly.
So that the granzymes and perforin those
Yeah. So basically, it's forcing that the only time that TC cell is going to perform its function is when it's in contact with an infected
and
cell.
that happens.
Yeah. Yeah. Devin and then Emma.
So just to clarify, the CD8 is the one that like joins together the TCR and the MHC1s.
The CD8 is the one that binds together the MHC1. Say that again. Sorry, I was losing track.
So the CD8 is the one that like contributes to the binding of the TCR and the MHC1. So that the
Yeah, it's almost as if there's two separate connections here. The TCR doesn't recognize MHC1. It recognizes the antigen inside or being presented by that MHC1. Does that help, Devin?
yeah, and then the CD8.
And the CD8's there to kind of hold this connection together. You can think of it as like additional support. Emma.
Doesn't all cells have like MHC1s? Like our T cells constantly binding and checking? Like,
Absolutely.
It's constantly happening, and then as soon as they find something for them, they'll act, but they just otherwise continue checking.
Yeah, now there's an additional step to this. Those TC cells have to become activated as well. And that's only going to happen if prior to this step, that TC cell has actually bound to MHC1 on a macrophage. And that's not something we covered in great detail. We did not talk about the activation. But there is an additional activation step that has to occur before this. Yeah. Go ahead, Zach.
So when these two things happen at the same time, it's destroying the whole MHC1 complex.
So it's not destroying necessarily the MHC1 complex. When these two things happen at the same time, it triggers... Oh, booger. There you go.
Thank you.
No problem. It triggers this cell to basically begin a process of death. So the ultimate goal here is that if this cell...
So that whole purple cell just comes out?
Yeah, and the reason, the benefit of that is if that cell's gone, it no longer can support the replication of the virus or the pathogen inside of it. Okay? This is actually one of the ways by which our body rids itself of mutated cells sometimes as well. A mutated protein would have a foreign structure to it. And so our body can recognize that foreign structure and kill those mutated cells. Abby, did you have a question?
Do TC cells always have the CD8 and the TCR on them? Like,
Yes.
are they already always equipped with that?
Yes, pretty much.
Okay.
For our purposes, yes, is the simple answer. Shall we go to NK cells? Okay. So sometimes MHC1 isn't on a cell surface. Most likely reason, a virus has inhibited the MHC1 from being produced or from being moved to the cell surface. We have a lovely backup system to deal with this. And that backup system are the NK cells. NK cells, we said, have two different types of receptors. They've got an inhibitory receptor and an activating receptor. Okay. If MHC1 is around, what you will get is the NK cell will bind, the inhibitory receptor will bind to MHC1, and then at the same time, the activating receptors, usually there's this activating molecule or other activating ligand on the surface of all cells as well. This binding will occur too. So these two things are happening simultaneously. But one force is much stronger than the other. Love the Star Wars connection, seeing as how we are only a week away from the new movie. I'm not Brenda, not into those movies as much.
What's the plus one called?
This would be like an activating ligand. So if this happens, the inhibitory signal is much stronger. In other words, if these two binding instances are happening at the same time, the NK cell's response is nothing happens. Which means a healthy cell, or if this were a pathogen, a part of a pathogen, what it means is the T cell would function. Okay? That would be the alternative.
Does it see cell?
These would kind of actually be occurring at about the same time. Yeah, good question. If though, oh, go ahead, Nicole.
What's the purple box?
Think of this as like, I just called it an activating ligand. Or an activating molecule. So it's on the surface of, it's like MHC1, it's presented on the surface of every single cell. It is there to recognize self in some ways, and the NK cell uses it additionally to bind to this self cell. Okay. Devin and then Danica. So this one is the activating receptor on the NK cell. Danica?
I thought all cells have the MHC1.
They do. They do.
Why isn't that on?
So there are cases in which MHC1 may not be expressed. One reason, a virus inhibits its expression. So viruses have a way to overcome that normal presentation. There's a benefit to the virus. That benefit is its antigens don't get presented on the surface of the cell. Okay? So it gives it a little bit extra time to survive. So again, if MHC1 is present, the NK cell does nothing. Nothing at all. If MHC1 is gone, though, that inhibitory interaction between this inhibitory receptor on the NK cell and MHC1, that interaction no longer exists. And so instead of this overpowering inhibitory effect on the NK cell, what we get instead is this overpowering activation of the NK cell. So it's actually the absence of the MHC1 that turns on the NK cell. When that happens, what does the NK cell do? Granzymes and perforin. Okay? Granzymes, perforin. Kills that infected cell. Emma.
So the T cell tries to bind, but it can't because the MHC1 isn't there, so then the NK cells are.
Yeah, that would be kind of the scenario, is that if so the TC cell tries to bind, there's no MHC1, it doesn't recognize it as self or non-self, it can't actually recognize it, so an NK cell would then be in charge of killing that infected
would
cell.
the TC cell bring the NK cell? Or like,
No.
It would just be like, how would the NK cell?
Cytokines.
Okay.
The simple answer is NK cells would be brought there by cytokines. All infected cells, and actually one of the mini lectures in the afternoon class brought up this term, it's called an interferon. Anytime a cell is infected, it actually starts to secrete what is called interferon. And the interferons are what recruit the NK cells. It's just another cytokine. Yeah, it's a really good question, though. Complexity. Okay, so that's how this process is occurring. Questions on this real quick? Or questions, or slowly. We can cover it slowly. All right, let's do one thing real quick. The only so this covers TC, NK. We've also talked about macrophages. We've also talked about neutrophils. The only ones I guess we haven't covered yet in our conversation here are B cells and T helper. Do we want to finish up with conversations of those before moving forward? Jamie?
Yeah, I also need like the diagram.
Yeah.
Should we know this stuff or put in there, or should
This is a different way to look at that problem. Whatever is simplest for you is probably the way that I would define it. However you logically think about it, okay? I would have concept mapped this, but I know you guys don't like the idea of a concept map. Most don't. I don't want to put everybody in that box. Abby hates it with a passion. Abby Sundberg. Maybe Abby Diedrich. All right. B cells and T helpers. Let's start with T helpers here. What do T helpers recognize? So, yep, MHC2. So let's go back to our presenter cell. In this case, we'll make it a macrophage. Macrophage is presenting MHC2 on its surface and presenting a piece of a pathogen with it. T helper cell, just like a T cytotoxic, there are two events that must happen at the same time. One, T cell receptor must bind to the antigen. Second, two, co-receptor must bind to MHC2. What is the name of said co-receptor? This is our CD4. Also, the molecular receptor for what virus?
HIV.
HIV. Talked about like 3 this semester. It's got to be one of those 3. Yeah.
Do both CD4 and the T cell receptor bind to antigen? What did that mean?
So it's the same kind of format. It mimics what we see with TCs. CD4 in this case is binding to MHC2, just like CD8 bound to MHC1, and then the T cell receptor is binding to the antigen. Okay. So those two events are happening at the same time. If they do, T helper becomes activated, and T helper does what? Cytokine release. So some of these cytokines would actually go back and activate the macrophage, make it more hungry for pathogen, increase phagocytosis. Some of those actually activate the T helper itself, autocrine signaling. And then the last would be to activate, and this, some of it might be to activate TC cells, some of it might be to activate B cells. So T helper controls a lot of the innate and the adaptive immune response all at the same time. Erica.
Which one's the innate? That's the B cell.
Innate would be, yeah, so let's break apart that term again. Innate are the ones that recognize patterns. Neutrophils, macrophages. Adaptive, change, recognize specific antigens. TCs, TH, B cells. Oh, you can throw NK under innate as well, because it recognizes a pattern. MHC1.
What goes on to innate?
Yeah, let me read. Okay. Single letters. B. T. Adaptive immunity. Innate. Innate to immunity. Neutrophil. Macrophage. And the NK cell. Because the NK cell isn't recognizing a specific antigen, it's recognizing a pattern. And that pattern is MHC1. Meg.
Can you just define again what neutrophils are compared to macrophages?
I can. Does anybody know? What neutrophil and macrophages are both types of what?
Phagocytes.
Both types of phagocytes. What is the difference between the two of them? Neutrophils come first, yes, that's absolutely true. What else is different? Which one of the two is a professional antigen presenting cell? Macrophages. If it's a professional antigen presenting cell, what does it, thank you, thank you. It presents MHC2. So one thing that neutrophils do not do, they do not express MHC2. They cannot communicate directly with Th or T helper cells in the same way that macrophages can. So there's our T helper. Go ahead, Mike. Yeah, I mean, that's one way to think of it. Since they're not professional antigen presenters, I mean, you can think the only role of a neutrophil is phagocytize. That's all it does. Eat, 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 eat.
So with that inflammation, macrophages are the first ones that are there.
So macrophages are resident to where that is occurring, and neutrophils are the first to be recruited to that site. B cells? Can we move on to B cells real quick?
So the macrophages are the ones that first transform phagocytosis because they're the first ones. They're like individually, but neutrophils are brought in first.
Yes. Yes. One thing I want to point out, don't get lost in all the detail. Okay? Don't get over vigilant with all the detail. And I'm just saying this because this is really confusing. I understand that. So if you would see what we tried to do last year with this section, you'd be a little thankful that we have really slowed this down. But don't get over involved in the detail, okay? That's my hope for you this Christmas season, holiday season. All right. I'm going to take this picture and I'm going to kind of redraw it because the event here between a B cell and a T helper is actually quite similar. There are some minor differences. How does a B cell recognize a pathogen? Before that. So in other words, does a B cell have a pattern recognition receptor or a PRR? No. What does it have? Well, it binds to a specific antigen. How? One way you could say this would be membrane-bound antibody. And actually, that's not an incorrect phrase or incorrect answer. Another way to think of this is an antibody, before it's secreted, before a B cell produces secreted antibodies, this is a B cell receptor, or a BCR. So T cells have TCRs, B cells have BCRs. B cell receptor binds a specific antigen on the surface, and then what happens? B cells are professional antigen presenting cells, so they must do what? What else can we associate with that term? MHC2, good. Let's work backwards on this then. So we know eventually what's going to happen is there's MHC2 with a bit of an antigen. What's got to happen before that? Phagocytosis. So B cells are professional antigen presenting cells. They must they will phagocytize this pathogen. That pathogen will go through the same process of breaking down as it happens in a macrophage. And eventually we get this presentation. And then the B cell is just showing off pieces of this antigen to any T helper cell that will talk to it. Okay? It's like that really annoying uncle at the holiday parties. or Zach. Not you. Me. All right. T helper, already having recognized this antigen previously on a macrophage, comes around and says, hey, I know what that is. Now what happens? Docking occurs, cytokines released. Now taking this B cell from kind of like a semi-active state to a fully active state, and what does that B cell do? He or she is the Oprah Winfrey of immune responses. Everybody gets antibodies. All right. That B cell begins, it's like, yeah, all right, let's party. And all it starts to do is hand out shots of antibodies to everyone. Okay, so that's the activation of the B cell there. So it requires it to not only bind to the pathogen and phagocytize it, but then ultimately for a T helper cell to come by, recognize that same pathogen and fully activate this B cell. Matt,
Could it ever be a T cell that goes to it?
you tell me. Could it why not?
Is it because it only has MHC2?
Yeah. If. And think about the pathway. So yes, you could actually get an interaction between a B cell and a TC cell. What would happen though? How would the result differ? What's a TC do? So you would actually get the death of the B cell rather than activation of that B cell. Yeah. This would be MHC2 here. And Matt, to answer your question, yeah. I mean, there are some viruses that replicate in B cells. Epstein-Barr virus is mono. Starts in epithelium, eventually moves to B cells later on in the course of an infection. And yeah, you will get death of some of those B cells that are harboring that virus. So yeah, it can happen.
So the difference between a T helper and a B cell is that in T helper, the antigen that was presented is actually killed off. But then in the B cell, like, I just don't understand. If it's the same cytokines released, why does one like the cell have the other?
It's not the same cytokines. We are just using the term cytokines generally to say that's what's being secreted. But there are different sets of cytokines. And one of the things that we haven't modeled in here is there's more than just these two interactions. There's actually a third and a fourth one involved. And those third and the fourth ones involved actually will tell this T helper which set of cytokines do I need to release. It's always dependent on who its partner is that it's bound to.
So what actually is the antibody? Like what is an antibody in relation to this?
So what is an antibody? It is a Y-shaped protein. So let's review the structure of an antibody. What's it do? How does it contribute to immunity? So, starting with this, which of the parts of this antibody is where an antigen would bind? What's that? Variable region. And where is that variable region located, Derek? Yeah, it's the arms. Okay? Remember, antibodies are really just trying to give hugs to pathogens. Big, lovely hugs. So those are the variable regions. There's two of them on each antibody there to bind to to an antigen.
You're using pathogen and antigen as the same thing.
I'm not trying to. I meant to say antigen here. I apologize.
So antigen binds to pathogen?
An antigen is a part of a pathogen. An antigen binds to an antibody. Antigen, side note, antibody generating. That's where the name comes from. Jesus has nothing to do with the naming. Yes. The B cell releases antibody. Well, the T helper's role is, as the name implies, it helps the immune response. The T helper, whatever cell it's binding to, if it recognizes the antigen, the T helper is just going to secrete cytokines to activate that cell. Okay? So then that cell is the one that's performing the function. Kaylin.
So the B cell releases the antibody attaches the antigen. So that's why the B cell activates.
Doesn't necessarily kill the antigen, it just binds the antigen. It's not the one that's actually doing the killing. Who's doing the killing? The macrophages. So a couple of things that antibodies do. One, they bind to an antigen. It inhibits that antigen then from actually being able to bind to host cells. Or sometimes we use the term it neutralizes the pathogen or the antigen on the pathogen. Okay. The other thing it does, it opsonizes it. It coats it. So if we have a pathogen,
What?
it coats the pathogen.
So it can no longer adhere to a virus or whatever it's going after. Yeah.
So here's the coating. Not coding, but coating.
Wait, hold on, this is the virus.
Virus,
Yes, yes.
antigen, antibody. Okay. Antibody is binding to the antigen. Antigen is part of the pathogen or virus. So the antibodies are coating the pathogen. That basically prevents this pathogen from doing anything.
How well do we need to know this?
It's like 11.5. So, well, you need to know the coding happens. It's called opsonization. O-P-S-O-N. Opson. Zation. Z-A-T-I-O-N.
What's the purpose of that?
What is the purpose of opsonization? That's part one. What else does this do? Anybody remember what this stem region is called other than stem region? The FC region. It binds to something. Binds to an FCR on the macrophage. Binds to an FC receptor on the macrophage. When that binding happens, what happens? We go all the way back to the beginning, somewhere, phagocytosis, all over again. In other words, a role of an antibody is to block a pathogen from causing illness by coating it, and then also to ensure that it gets eaten and destroyed. All right?
You're not going to make us connect this to immunizations, are you?
To what?
Immunizations.
Oh yeah, we have to. Because what is immunization? What does a vaccination do? It produces what? Antibodies and immunological memory. memory B cells, memory T cells. So yeah, you kind of have to connect it because they are really one and the same. Vaccines are just applied immunology. Okay? Okay. All right, I'm going to pause for a minute. Give everybody a chance to breathe. Paige? Hanging in there?
Memory T cells and T cells and liver cells.
It depends.
Depends on the vaccine or the.
Depends on the type of vaccine. Jamie? No, that was to go through specific examples of pathogenicity factors. That is something that I would like you to know about. Her question was about my beautiful anatomy picture of the epithelial cells and the epithelial cells that make up the streptokinase or streptomoniae. Well, the coding is all the way right there. Oh.
That one, yeah.
No, absolutely not. And that's a leftover question.
Where does the variable region bind to macrophage again?
So it's not the variable region that binds to the macrophage. It's this FC or this constant region. So it binds to macrophages also, in addition to the PRRs, have FC receptors,
Okay.
which allow them to bind to these antibodies.
FC receptor.
Okay.
I'm gonna go over at some point what we did last Friday and stuff like that. Anatomy that you didn't talk about.
Yeah, we can go over some of that. Okay. I'm going to pause. Anybody have any other questions on this stuff?
What? I'm sorry.
No, go ahead.
The FC region's on the antibody, but the FC receptor is on a macrophage.
Yes. Yes. C. All right.
Zach, when do we have to do the first email now?
Sunday. You know what? I did get another link sent to me. I was going to send it out to you. But otherwise, I got an email when I asked about it, and Linda Wilder said email her if you still can't get to it. But will you remind me tomorrow in lab, and I will, if I haven't done it tonight, I will send it
Yeah,
out.
because another teacher's giving us a credit, and I'm like, I want to do this.
Erica? No. No. You need to know how we test them, okay? And how to measure effectiveness. Yeah, period. Period. Okay, so I was asked, hey, what do we need to know from Friday?
When you were here, and I was not meant to be here.
Oh. Okay, so from last Friday and from Monday, I don't need you to know the specifics of strep pneumoniae. I was using that as an example to try and create some sort of personal connection to the various things that we talked about. So strep pneumonia is a good example. And if you want to put this into some sort of context, go back over that stuff. But the importance is not to remember CBPA as a protein that's involved in attaching to certain types of receptors on the host. Rather, it's the general theme. If a bacterial protein attaches to a receptor on a host cell, we can define that bacterial protein as a what? And it's one of these terms up here. It's an adhesin. It helps the bacterial cell to adhere to host cells.
Is it just for bacteria or viruses?
It can be for viruses. It can be for parasites. It can be for fungi.
And so the bacterial protein that attaches to what?
So it allows the bacterial protein to attach to host cells. Okay? Or we did talk about a couple of examples that help strep pneumoniae attach to the extracellular matrix. So it basically allows for the bacterium to adhere and to colonize inside the host cell, or inside the host. What are invasins? Meg?
Enzymes that allow for invasion of cells and other tissues.
Enzymes that allow for invasion into cells and other tissues. One example we said was hyaluronidase. And this was one that breaks down the hyaluronic acid. There are other ones that help to break down components of the extracellular matrix as well, allowing the organism to get out of a tissue or an organ and into a new system. Bloodstream, for example. Okay. Adhesions and, sorry, go ahead.
For adhesions, is it inside the host cell?
It doesn't,
Outside.
outside. But we can have, and this kind of takes me to the next point, adhesins and invasins can be one and the same. So for example, if the adhesion of a bacterial cell triggers endocytosis, bringing the organism into the cell, that then classifies it as an invasin. So we said, for example, the CBPA on strep pneumoniae allows for strep pneumoniae to not only adhere, but once the binding occurs, it triggers endocytosis and allows it to migrate through that cell to the other side. So that would be an example of an adhesin that is also an invasin. I don't think you need to know that specific example. I think knowing that an adhesin and a protein can be both of them is an important concept. Just so that you don't, I just don't want you to think that it has to fit nicely into one or the other category. It can be both. Devin, did you have a question? Okay. Function of a capsule, at least in context of immunity, immune system. Not penetrated, but it prevents the phagocytosis. So capsules, if you'll recall, are a thicker coat of sugar outside of the cell wall. We described it kind of like a cloaking device, in that the thickness of it will prevent the pattern recognition receptors on the surface of a macrophage or a neutrophil from being able to even come into contact with some of those PAMPs. So it prevents the phagocytosis. Mike?
So would we consider that like good for the fat thing, but like bad for humans?
Absolutely. Absolutely. Go ahead.
Something about the capsule help it pick up like the IgAs or like the info?
Oh, gosh. So,
Or is it just like
it's not so much that it helps it to pick up the IgA, it's that it would actually prevent the IgA also from binding to specific antigens within. The IgA may be able to bind to the capsule, though. That's a different story. But it would prevent detection by IgA. Yeah. We've brought up many other examples, okay? Now, is it possible that I'll give you something on the exam that you haven't seen? Absolutely, okay? But I would provide you context. So, for example, let me make up a protein name. Okay, Cryptochrysma-Quanzal lysine. It's a secreted protein. Produced by the gram-positive organism Santa Nicholas
Just go with that.
reindeerus. Okay, so gram-positive organism by that name. It's a secreted protein, and actually, when you mix it with macrophages, the macrophages, they go from this to they actually explode. Okay, so what is this protein? How can we classify it? Good. It's an exotoxin. We know it can't be an endotoxin. Why? It's from gram-positive. The other thing we know is endotoxin is a lipid and not actually a protein. Okay? How else can we classify this? What kind of potential exotoxin might this be? Or what might be similar to something that you've heard about?
Like red blood cells, how they
Good. What was that called? A hemolysin. In this case, we could just classify it as a cytolysin. This one, I suppose, technically would be a leukolysin or a leukocidin because it's killing white blood cells. But it's a lysin, right? It will lead to the disruption or explosion of those cells. Yay. So, and this is something that I have tried to reiterate since day one of the semester with all of this material. I could make up the most ridiculously stupid question on the face of the earth. But can you put this into the context of what you know and what we have talked about? Given the context of this problem, yes, you should be able to answer what type of molecule this is. Meg?
So would like the license, would they all be exotoxic?
Yeah. Yeah, they would.
So that's how you would also know it because it's lysine,
It-
and you know it'd have to be an exotoxin?
Yeah. That would be a perfectly fine answer, given what we've talked about. And I don't know of an exception to that. That's a great point. What are some other things between exotoxins and endotoxins that we know? So we know that we've said that exotoxins are secreted. Obviously, endotoxins are not. We've said gram status for exotoxins. It can be from a gram positive, but we know that endotoxins only come from gram negatives, right? Exotoxins could be gram negative, gram positive, mycobacterium, other. What else do we know about these two in comparison to each other? Devin? Which ones are? Endotoxins are heat stable. Exotoxins are not heat sensitive. Therefore, that allows for exotoxins to be used as what type of vaccine? What type of vaccine is produced from an exotoxin? Those are specific examples, yes, but category-wise,
Or something.
subunits, but more specifically, it's one that actually has tox in the name. Toxoid. Toxin. I couldn't hear you, Jamie. I'm sorry. Didn't you have to leave at 5:32? Okay. All right. So toxoid vaccines are produced from toxins, and we can do that because they are not heat stable. Because once we heat them, they will denature, but they still retain their antigenicity or ability to trigger production of antibodies. Other features, I think those are the major ones. Which one is a stronger antigen? Exotoxins are stronger, takes less of it to cause a response. Both of them we said that can cause one side effect of either of them because they can trigger inflammation would be the ability to cause shock as well. Okay. With, exotoxins and endotoxins both can. Meg, did you have a question? Oh, sorry, Olivia, you did.
For an endotoxin, does the cell have to lyse?
That's a great point. Yes. So the gram-negative cells actually have to lyse or be destroyed in some way for that endotoxin to be released. Absolutely. That's a really good point. Go ahead, Mike.
Can I ask a question on that?
Sure.
For that table that we had to fill out
Yeah.
today, what do we all have to know?
Afternoon class?
All of it.
You need to know all of it. Okay. So basic is you need to know the general information. And I know we didn't spend a lot of time covering it today, but honestly, that general information is all in the mini lecture. I am going to put a key up for it, first of all, and I will try to get to that tonight. By the latest, it will be up as tomorrow morning. Okay. But the general things that I would like you to know. So there are 6 different types that we talked about, and that's not necessarily important, but. And I'm going to group them real quick, and it may become apparent in a minute why I'm grouping them this way. I'm going to just put DNA down there for now because, to be honest, I really wish we had more time to talk about this one. We don't, so I would say let's just take that list of 6 and drop it down to 5 immediately. Okay? Don't worry about the DNA ones. They are the one thing that I usually tell folks, and I didn't have the opportunity to today. We've been talking about DNA vaccines for since the time I was an undergraduate. Actually, one of the pictures that I have on the slides, I think might have been one of the same ones that my micro professor showed me back in the day. The idea of it hasn't changed. We are really no further than we were 15 years ago with it. That's maybe not giving it enough credit, but we don't have a DNA vaccine just yet. These are the major ones, and I've kind of grouped them this way intentionally. So all of these ones right here, toxoid, subunit, and conjugate, what are some common features of these ones? Perfect. They're parts of the pathogen. Sorry, I'm just emphasis. All right, they are parts of the pathogen. So subunit is kind of the broad class, and then there are these 2 subclasses, toxoid and conjugate. Subunit, part of the pathogen, oftentimes produced in a bacterium or yeast or something like that. Can these cause disease? No. If prepared correctly, cannot cause disease. HPV vaccine is a subunit vaccine. It is made up of 2 parts of the virus that makes up the capsid structure of it. Contains no DNA in it, so it therefore cannot replicate. And actually, that kind of takes us to the next point. What thing or things do these four have in common? They're all dead. Can they replicate? No. In other terms, what arms of the immune response, what adaptive immune cells can they activate? Perfect. These all can activate B and T helper. We get a really robust antibody response to these cells, or to these types of vaccines. What else might be true about these?
You have to get another one.
A booster.
Yeah.
Perfect. We have to retrain our system sometimes. Okay.
So like the memory cells aren't as strong as.
Yeah. Our memory cells necessarily aren't as strong as they would be with something that is attenuated, for example. How, what's common between inactive and attenuated? It's the whole pathogen. Perfect. And then finally, what makes attenuated unique from the other four? Replicates, stronger immune response. What were you saying, Jenna? Okay, perfect. What else? No booster. What's that? Yeah, and I guess I'm not as worried about the refrigeration part. I think that I'll be honest in saying I don't exactly remember what I said on the mini lecture for parts of the refrigeration here, because there are certain ones even that like Kathleen had brought up today that are subunit ones, and if we want to retain the three-dimensional conformation of the protein, it needs to be refrigerated. Okay, so even that can be a little bit iffy depending on the vaccine sometimes. But what is true about the immune response that is generated here? Lifelong, typically. There are exceptions to that, or we think there may be exceptions to that. I guess I should put.
Do you know the difference, like, more RNA?
Oh God, that's a good question. No, I'm not as worried about that, but that's a great topic for another time. She was asking, do you guys need to know that this is more common with like attenuated viruses, or vaccines are more common for RNA viruses? And the answer is no. Well, it comes down to the mutation frequency, right? RNA viruses are easier to attenuate because they mutate and adapt to a new condition a little bit faster. That was something we talked about in the morning, not in the afternoon today. Derek?
You mentioned that these would be spread.
That's a really good one. Attenuated can be spread from one individual to another. Absolutely. And we saw that with poliovirus as a great example. Yeah, through poo-poo, right, exactly. Swimming pools, right?
Get you.
What was that?
What do you mean when you just say spread from one person to another? Just like the virus or what?
Yeah.
Oh, okay.
So like polio is passed through the oral fecal route, replicates in the stomach or in the gastrointestinal tract. As soon as somebody poops it out, if somebody comes into contact with that fecal matter in some way, shape, or form, and then like, I think children, okay? So child steps in poop, infected with polio. Meg?
And that would stimulate both B cells and TC and TH?
Yes, stimulates all three. And that's probably one of the, and that's why we don't require a booster, and that's why it triggers such a strong lifelong immunity. Devin and then Paige.
Is that, when you were talking about like people and stuff like that, is that kind of like the natural and active pathways?
Yeah, it kind of is. And that's a little bit different. I mean, like, I could set up that kind of question with this, but the last part of the immunity that we talked about today was know the difference between the four categories: natural active, natural passive, artificial active, and artificial passive. I would say you probably want, maybe one of the easiest ways to kind of memorize these is come up with an example for each. Is one of the easy ways to do it. Okay. Paige?
So like, you know how some people can get chickenpox like twice?
Yeah.
You're only supposed to get it once.
Yep.
Is that just like a failure of your memory cells? Or is it due to like mutations of the
In the case of chickenpox, it's probably either a failure of your memory cells, or the second possibility is you're immunocompromised. And so your immune system which I think kind of goes hand in hand, but sometimes it's not that the memory cells are gone, there just maybe aren't as many of them. And so it gives the response is slower. Yeah, that's probably one of the ways to describe it. Yeah, great question. In the case of chickenpox, it's probably not a mutation because it's a DNA genome. Cold, on the other hand, well, there's over 100 types of cold, which is why we're not going to get rid of them. Anything else? I will hang around for a few minutes, but I will hang around for a few minutes. I'll just leave that there. Yeah, yeah.
So artificial non-active,
Artificial active?
inactive.
Passive.
You have the IV. Is that like IV? Like if you're getting ceftriaxone or something, IV antibiotic being used in maybe because. Does that make sense?